that across. So it helps to regulate the fluidity of the plasma membrane. As we continue to take a look, we have the major functional component of the plasma membrane will be that of the proteins. So the major structural component, we can identify the phospholipids, and then we can highlight the proteins as being the major functional component. So we have the proteins, the third component. And these are membrane proteins. These proteins are a major component of the plasma membrane. So that major functional component of the plasma membrane. As we take a look, we have two types of proteins, or two classes of proteins associated with the plasma membrane. They're called either what? Integral proteins or peripheral. So we have two types, two classes. We have the integral proteins and the peripheral proteins. When we say an integral protein, what do we mean when we say an integral protein? Right, so the integral proteins are what? Embedded within the what? Plasma membrane. So these proteins will interact with the hydrophilic of the plasma membrane. So we think about those amino acids that form a portion of the protein that interacts with the hydrophobic core. These must be those non-polar amino acids forming that structure. So as we think about the integral proteins, they're embedded within the plasma membrane. Most of the integral proteins are classified as being transmembrane proteins. So large of the integral proteins, which are embedded within the plasma membrane, interact with the hydrophobic core, are called transmembrane proteins. When we say transmembrane, they span the thickness of the plasma membrane. They have both a what? Extracellular phase and a intracellular phase. So most of the integral proteins are transmembrane, where they actually span the thickness of the plasma membrane, exposed to both the extracellular side and the intracellular side. And we're saying the proteins, the amino acids that form the portion of the protein that can interact with this hydrophobic core. This would be a sequence of the non-polar amino acids. So we have a non-polar side chain. Think about the functions of the integral proteins. Make sure you take a look at the table or the figure in your textbook where they highlight, so we have 6 or 7 different functions of the transmembrane or the integral proteins of the plasma membrane. For example, they can function as transport proteins. We have several different types of transport proteins. We'll talk about membrane transport, but we can have channel proteins. So we have channel proteins that allow ions to either enter or exit the cell, because we said these substances are charged. They cannot really interact with the what? Hydrophobic core of the plasma membrane, or they can be, this is a channel protein, or we have what are called carrier proteins, which function as proteins. They have a specific binding site that bind to a particular solute to transport that solute across the plasma membrane. So we have transport proteins. We have what are called CAMs, or cell adhesion molecules, which allow adjacent cells to form cell junctions, to link cells together to form our tissues. Then we have receptors. They can function as receptors. we say receptors, correctly, what's the major function of receptors? What are they involved in? They bind to what? Ligands. We say a ligand. What can be a ligand? A hormone or a neurotransmitter. So the receptors involved in our cell communication bind to chemical signals. So they bind to chemical signals. When we think about the insertion of the receptors in the plasma membrane, the phospholipids around the region of the plasma membrane that has inserted into it has a high concentration of saturated fatty acids, and the region of the plasma membrane where we have these receptors, where we have a higher concentration of saturated fat, are called lipid rafts. As you know, when you say lipid raft, this is just a region or a patch, so to speak, of the plasma membrane where we insert these receptors, which are involved in cell-to-cell communication, in the molecular profile of these phospholipids, and we say they have a high concentration of saturated fatty acids. If this is the case, what can we say about the nature of the plasma membrane at the lipid rafts where we insert these receptors? What can we say about the nature or the consistency of this region of the membrane? If they have a high concentration of saturated fatty acids, what can we say?
It's going to be less fluid.
It's going to be less fluid-like. So stabilize the positioning or the anchoring of these critical in the plasma membrane. We have a higher concentration of saturated fatty acids, so it's less fluid-like. And this helps to anchor or secure the positioning of these proteins in the plasma membrane so they can effectively what? Bind to chemical signals to regulate the activity of the cell. Okay with that? Then we have the peripheral proteins. The peripheral proteins are within the plasma membrane. They do not interact with the hydrophobic core. Not embedded within the plasma membrane. They're loosely associated with the integral proteins, and the peripheral proteins will either be exposed to the extracellular fluid or the intracellular fluid. They're not transmembrane proteins, so they're loose. We have a weak association between the integral proteins and the peripheral proteins. They're exposed either to the extracellular phase or the intracellular phase. They can function as enzymes. Peripheral proteins. So make sure you take a look at the functions associated with the peripheral proteins, but clearly structurally, not embedded or not transmembrane proteins interacting with the hydrophobic core of the plasma membrane. And then the last component will be our carbohydrates. These are not carbohydrates involved in energy to the cell, but we have our carbohydrates. These are short chain polysaccharides that will complex with a protein or a lipid associated with the plasma membrane to form our glycoproteins and the glycolipids. Complex with lipids and proteins associated with the plasma membrane to form the glycoproteins, to form the glycolipids. The carbohydrate component of the plasma membrane is asymmetrical. It's only exposed to the extracellular phase. Of the plasma membrane. Each cell will have a sugar coating, so to speak, on its extracellular phase. So on the extracellular phase of the plasma membrane, every cell, we have these carbohydrate moieties complex to the proteins and the lipids to form glycolipids and glycoproteins. they form what is called the glycocalyx. And the glycocalyx will help identify particular cell types, and the glycocalyx will play a key role in cell-to-cell communication, in embryonic development for tissue formation. The glycocalyx of epithelial cells will be similar of all epithelial cells and allow epithelial cells or like cells to form cells to form our what? Tissues. So it plays a key role in cell-to-cell communication and recognition, pivotal to tissue formation. What I'm trying to say here is the glycocalyx for an epithelial cell will be different than the glycocalyx of a what? Muscle cell or a nerve cell. Allows like cells to recognize like cells, which is pivotal to tissue formation. Are we okay with that? Also, the glycocalyx will be used by our immune cells to recognize what? Bacteria and foreign materials. So it also plays a key in immunity and recognizing foreign material. So our macrophages will recognize a glycocalyx bacterial cell identified as being a foreign cell, mount an attack against that cell, and destroy it based on the profile of the glycocalyx. So make sure you're familiar with the four major components of the plasma